Okay, so welcome to this next video in which we are discussing endocytosis, specifically Clathrin Mediated Endocytosis. And I should stress that there are different mechanisms of endocytosis, but we're specifically discussing Clathrin Mediated Endocytosis, which I think it would be fair to say is one of the most important forms, maybe. Okay, so phosphatidate. We're now discussing the process by which we actually uncoat our clathrin-coated endocytic vesicle, and we've discussed that endophilin dimers, which are still associated with the clathrin-coated endocytic vesicle, are important in recruiting the phosphatase synaptojanin. Now, I've told you that synaptojanin is going to act on PIP2, and to understand what it's going to do to PIP2, we need to know the structure of PIP2, and to understand the structure of PIP2, I've firstly shown you the structure of a normal old phospholipid. So, let's now look at PIP2 and see how it's just basically a modified phospholipid. Okay, so let me draw you the phospholipid again, and we will then modify this phospholipid to create PIP2. Okay, so here's the glycerol in orange, in green, here are the fatty acids in orange, and here is the phosphate group in red here. Okay, so PIP2 then. PIP2 is short for phosphatidyl, and now you see why it's important to know what a phosphatidate molecule actually is. Phosphatidyl inositol, and that inositol really should be connected to the phosphatidyl, so let's put a little slash there. Inositol, 4,5-bisphosphate. Okay, and the reason this is denoted PIP2 is that the first P stands for phosphatidyl, the I stands for inositol, and the last P stands for phosphate, and then because it's bisphosphate, because you've got two phosphate groups attached to the phosphatidyl inositol molecule, you put P2 there. Now, we have a phosphatidate molecule. This is the phosphatidyl portion of this name. Phosphatidyl basically just means that you have stuck a phosphatidate molecule onto an inositol molecule. Okay, so basically, to turn this into phosphatidyl inositol, what you do is you stick an inositol ring off the phosphate group of the phosphatidate molecule. Now, what is inositol? Because inositol is one of those molecules that generally no one has a clue what it actually is. Inositol is, if I draw its skeletal structure, it's a beautifully symmetrically rotational molecule. Okay? Rotationally symmetric molecule, rather, not symmetrically rotational. Okay, so here's our skeletal structure. So it's a 6-membered carbon ring, okay, where all of the bonds within the carbon ring are single bonds. And because we're drawing a skeletal structure, it means I'm lazy. I don't have to draw the actual carbons. Instead, these corners mean a carbon, okay? Now, where do each of these carbons get the other two bonds they need from? Well, basically, every single carbon also has an alcohol group or a hydroxyl group sticking off it. So here are these alcohol groups coming off every single one of the carbons of the inositol ring. Okay, now that gives all the carbons 3 bonds. Where do they get the 4th bond from? Well, they also have a hydrogen atom stuck off each one of them. So that's the structure of inositol here. Right, so in my cartoon of PIP2, I will just denote inositol by this blue hexagon here. So this is the inositol ring. So that's now phosphatidyl inositol, because I've got an inositol group with a phosphatidate or a phosphatidyl group stuck off it. And then we want phosphatidyl inositol 4,5-bisphosphate. So what we need to do is add phosphate groups onto the 4th and the 5th carbon of this inositol ring. Now, the way you name the carbons of the inositol ring is you name this one that's actually attached to the phosphatidyl group. You name that carbon 1. Then you go around rotationally. So 1, 2, 3, 4. Oh, whoops, there's 4. And let's stick a phosphate group onto it. 5, let's stick a phosphate group there, and then 6 up here. Right, so that now is phosphatidyl inositol 4,5-bisphosphate. Okay, right. So, what we now want to do is see what synaptojanin is going to do to this. But before we do that, let me just make some comments on this molecule. Hopefully, what I have convinced you is that this is just a phospholipid with a bigger head. Yes, it's got this extra bit stuck on, but it's just a normal phospholipid. And indeed, you can put this in the phospholipid bilayer and it will function just like a normal phospholipid as far as the membrane function is concerned. So, when we endocytose some membrane, when we make our endocytic vesicle from the plasma membrane of the cell, then we're going to end up with PIP2 molecules in that endocytic vesicle membrane. Okay, so synaptojanin, which we've now recruited to our endocytic vesicle via the endophyllin dimers, will now act on these molecules that are within the endocytic vesicle. And what it's going to do is it's going to break it down, basically. Now, what's it going to do? It's not going to do a massive alteration. Instead, I told you it was a phosphatase, so it's going to remove one of these two phosphate groups. Now, you've got a 50-50 chance. It's actually this one, this fifth one here. Okay, so you remove that one phosphate group off the inositol ring. Now, what do you get? Well, you're going to get the whole molecule bar that phosphate group. So you've got your phosphatidyl here. You've got your inositol. Okay, so here's your inositol. You've got your phosphate off the fourth, and then you haven't got a phosphate off your fifth. And hopefully, you should now be able to work out what this molecule should be named. So let me colour in all the bits. So here are the long-chain carboxylic acids. Green, we've got the glycerol. In red, we've got these two phosphate groups. And then in between, we've got the inositol ring here, this six-membered blue ring. Now, what is this structure here? This is phosphatidyl, because you've still got this phosphatidate molecule up here. So phosphatidyl, and then we've got inositol, so it's phosphatidyl inositol, and then we've got one phosphate off the fourth carbon, so it should be 4, and then phosphate, because we've only got one phosphate, so we don't need bisphosphate anymore. So this is phosphatidyl inositol 4-phosphate. And for short, phosphatidyl will be P, I for inositol, so PI means phosphatidyl inositol, and then we've just got one phosphate, so this is PIP, PIP. Okay, so what happens is when we recruit this synaptojanin to our endocytic vesicle via it binding to the endophyllin, what's going to happen is synaptojanin is going to start breaking down the PIP2 or PIP2 into PIP or PIP. Okay, and when this alteration happens, that is what triggers the removal of the Clathrin coat. Okay, so now the Clathrin coat is going to be taken off. Okay, so here we now have an endocytic vesicle that has no Clathrin coat. So we now want to see what happens to this endocytic vesicle. Where does it go next? And the pathway which now happens to this endocytic vesicle, so here's the plasma membrane, which I'll just label as PM for plasma membrane. The pathway that now happens to this endocytic vesicle is what's known as the endocytic pathway. Okay, so do not confuse the endocytic pathway with Clathrin mediated endocytosis. Many people will be inclined to think that the endocytic pathway means that entire pathway which we've discussed now, the pathway of Clathrin mediated endocytosis, but in fact the endocytic pathway is what happens next once you've actually endocytosed your endocytic vesicle. Okay, and we'll discuss the endocytic pathway in the next video.